chemical structure and base composition of DNA and RNA, DNA and RNA, DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are the two primary types of nucleic acids, essential biomolecules responsible for storing, transmitting, and executing genetic information in all living organisms. These molecules act as the blueprint for the synthesis of proteins, which are critical for the structure and function of cells. DNA is the hereditary material found in almost all cells of living organisms, carrying genetic instructions necessary for growth, development, and reproduction. In contrast, RNA plays multiple roles in gene expression, primarily in the synthesis of proteins from the information encoded in DNA. Both DNA and RNA are polymers made of nucleotide units. Each nucleotide is composed of a sugar, a phosphate group, and a nitrogenous base. Although they share a similar basic structure, they differ in key aspects like the type of sugar, base composition, and structural form, allowing them to perform distinct biological functions. Understanding the chemical structure and base composition of DNA and RNA provides insight into their roles in genetic processes, replication, and protein synthesis, forming the basis for fields like genetics, molecular biology, and biotechnology. Chemical structure of DNA. DNA is a polymer made of repeating units called nucleotides. Each nucleotide consists of three components. Deoxyribose sugar, a 5-carbon sugar, pentose. In DNA, the sugar is deoxyribose, meaning it lacks an oxygen atom on the second carbon, hence the name deoxy. Nitrogenous base. There are 4 nitrogenous bases in DNA, divided into 2 categories: purines, double ring structure, adenine, A, guanine, G, pyrimidines, single ring structure, cytosine, C, thymine, T, phosphate group. A phosphate group is attached to the 5' carbon of the sugar. The phosphate groups link the nucleotides together, forming the sugar-phosphate backbone of the DNA molecule. Structure of the DNA molecule. Double helix. DNA is a double-stranded molecule. The 2 strands are coiled into a helical shape, antiparallel strands. The strands run in opposite directions. One strand runs from 5' to 3, and the other runs from 3 to 5. Base pairing. Bases on opposite strands are paired by hydrogen bonds. Adenine, A, pairs with thymine, T, with 2 hydrogen bonds. Guanine, G, pairs with cytosine, C, with 3 hydrogen bonds. Complementary base pairing. This pairing ensures that the 2 strands are complementary, meaning the sequence of one strand determines the sequence of the other. Base composition of DNA. The base composition refers to the percentage of each nitrogenous base in a DNA molecule. According to Chargaff's rule, the amount of adenine, A, is equal to thymine, T, and the amount of guanine, G, is equal to cytosine, C, Ag, purines equals Ct, pyrimidines, RNA, ribonucleic acid. Chemical structure of RNA. RNA is also a polymer of nucleotides, similar to DNA, but there are key differences. Ribose sugar. The sugar in RNA is ribose, which has a hydroxyl group, OH, attached to the second carbon. Nitrogenous base. RNA contains 4 nitrogenous bases, but instead of thymine, T, it has uracil, U, purines, adenine, A, guanine, G, pyrimidines, cytosine, C, uracil, U, phosphate group. Just like DNA, the phosphate group links the nucleotides together to form the backbone of the RNA molecule. Structure of the RNA molecule. RNA is typically single-stranded, but it can fold into complex 3-dimensional shapes due to complementary base pairing within the same strand. The different types of RNA include mRNA. Messenger RNA carries genetic information from DNA to ribosomes. tRNA. Transfer RNA helps in the translation of mRNA into proteins. rRNA. Ribosomal RNA, a structural component of ribosomes. Base pairing in RNA, adenine, A, pairs with uracil, U, guanine, G, pairs with cytosine, C. Key differences between DNA and RNA feature sugar, DNA deoxyribose, RNA ribose, feature strands, DNA double-stranded, usually, RNA single-stranded, feature nitrogenous bases, DNA adenine, A, thymine, T, cytosine, C, guanine, G, RNA adenine, A, uracil, U, cytosine, C, guanine, G, feature base pairing, DNA AT, GC, RNA AU, GC, feature function, DNA genetic information storage, RNA protein synthesis and gene regulation. Diagram showing key differences between DNA and RNA. Thank you for watching.